by the Indian Agricultural Research Institute, sponsored by NAHEP CAST. This workshop marks an important occasion as we come together to exchange knowledge, ideas, and experiences in the field of plant microbiome research. The plant microbiome is an incredibly fascinating and complex area of study, and it is crucial to enhance our understanding of the topic and promote collaboration.
NHP cast, the distinguished experts, Professor Jos Rijmakers, with the Director Microbial Ecology, Netherlands Institute of Ecology, and Professor Leiden University, the Netherlands, Dr. Victor Carrion, Dr. Guillermo, Dr. Lucia Maria, Dr. Vishwanathan, the Joint Director Research and Project Principal Investigator, NHP CAST, Dr. A. Kumar and Dr. B. Ramakrishnan, the course coordinators. Large number of heads of the division, professors, faculty members are attending this function online. I welcome all of them. And most importantly, the participants are the student trainees who have been earned their seat because they were the lucky ones among, I think, 1:5 ratio. We received their applications. So you have got the opportunity to get the trainings on this workshop. So I welcome all of you for this inaugural function of the International Training Workshop on Plant Microbiomes, Theory and Application, which has been scheduled from today onwards till 3rd March 2023. The objective of this workshop is mainly to train the students and young faculty on the emerging area of plant microbiomes, microbial genomics, and their applications in agriculture with special reference to relevance to the crop protection and management. As a customary, I, on behalf of the Institute, the NHEP, and the CAST team, and all those present online and physically present here, I welcome the dignitaries and I request Dr. Vishwanathan, the PI and Joint Director Research, to do the honor of presenting the bouquet of flowers to our dignitaries from overseas and our own pillar of NHEP respectively, that is Professor, Professor Jos Rijmakers, Professor Jos Rijmakers, course director. And Dr. Anuradha Agarwal, she is one among us and she is the supporting person for anything which happens at this center. Now I request Dr. Kumar and Dr. Ramakrishnan respectively to welcome our two more dignitaries. We have come from Leiden University, Dr. Victor J. Carrion and Guillermo and Lucia Maria. Dr. Ramakrishnan, please. Yeah. One more. Now I request again Dr. Kumar to present a bouquet of flower to our pillar of this NHEP CAST IR in New Delhi, Dr. Vishwanathan. Before I hand over the mic, it is my privilege to, please have seats. It is my privileged duty, and it is a customary in Indian mythology. Everything we start with lighting the lamp. So light is the source of inspiration to unravel the unseen, to ravel the unseen, and also to excavate, which is we are not sure about. So to make this function fruitful and effective, and the way we want to conduct this function, I request all the honorable dignitaries join hands for lighting the lamp ceremony. Thank you very much.
Thank you, sir and ma'am. Now I request Dr. A. Kumar to brief the audience about the program.
Very good morning to all of you. It's a great occasion for IRI, for the Discovery Center, for this project. We have been working hard on this particular project, more so for inculcating the knowledge in IRI students on the frontiers areas of science. That includes genomics. Genomics includes metagenomics. Metagenomics also encompasses the microbiomes. So we wanted to have this project, this particular training program for the benefit of IRA students. And we have been working for the last 6 months. And the foundation for this particular training workshop was shown in Vienna when I met Professor Icebreaking. We happened to interact quite a lot about microbiome projects, what's going on in India, in Netherlands. Then he readily agreed. Thank you, Professor Icebreaking, for accepting our invitation to be here to train all of you. And all of you must be knowing, Icebreaking's group is one of the leading groups in the world working on plant microbiomes. You can see their publications, their achievements, their accomplishments in several high-end publications in the world. I never thought anybody other than Roy Makers to be a resource person for this particular training program in IRI. We have structured this program and we have given the entire responsibility to conduct this program to the team led by Professor Roy Makers. And he brought an expert in the area of different domains in microbiomes. We have an expert for metabolomics, we have an expert for computers, we have an expert for bioinformatics, and I don't want to reveal what they are going to do here. I leave the floor to Roy Makers to highlight what they have in their mind to teach all of you about this microbiome research and how this microbiome can be harnessed for the benefit of agriculture. So with this brief note, I call upon Roy Makers to be here just to highlight what we have in our mind and what is there in store to communicate to all of you, to teach all of you. Roy Makers, please.
Okay, good morning. Good morning to everybody. It's a real pleasure to be here again. So this is my third time to India. But I've never traveled north. So I traveled only south of Bombay. So this is a totally new experience for me, I have to say. The diversity in this country is enormous, and it's a super pleasure to be exposed to that. So, and during this week, two weeks, I'm sure that you will learn also us more about your culture, and so please do so. We are open to any new experience. So it's going to be the course is going to be a two-direction, interaction. So so I hope to learn also from you. Thanks also to the organization, also to the funding bodies that made this happen. I think this is a unique and beautiful opportunity to interact and to learn from each other. We're going to teach you also, of course, on microbiomes. Thanks, Kumar, also for initiating this. We know each other for quite a while. We first met in 2003, 20 years ago. And that was at a meeting, a PGPR meeting in Kerala. So that's where we first met, and then since ever we are still in contact. We mail each other often, exchange ideas, and thank you so much, Kumar, for this opportunity. Also to my team, I will introduce them in a minute on the slides. Shall we? Yeah, so this is of course the title.
Can we go on?
So this is, and I'm sure that I missed, because I did not have pictures of all the people that contributed to the course, right? So forgive me for that. It's the ICAR, IARI team here. I met people on Saturday. They've been super busy preparing everything for the lectures, the practicals. Thank you so much for organizing this. Also, of course, like I said, there's many people behind the scenes who made this course happen in terms of funding, in terms of organization. So many thanks. I put this slide up because this will be the people that you will be interacting with in the next 2 weeks on a daily basis, right? And I just I tried to pick some pictures from the internet. Dr. Ramakrishnan, of course, well-esteemed microbiologist. In terms of work, I've seen many things, also antibiotic resistance, but biogeochemistry, that was a very clear signature there. Of course, Dr. Kumar here, plant pathology and plant microbiome research as the specialty. Then I want to introduce, so that this is a picture we took yesterday with Vinod, who was an excellent host to show us around, also in Old Delhi. Beautiful. We enjoyed a lot. So thanks, Vinod, for doing that. So Dr. Victor Carrion here, present. Maybe you can just stand up a bit, Vic. Yeah, so Vic. So we try to bring also from our institutes a quite diverse expertise, also in terms of age distribution. You know, we took care of that. So going from young to medium age to me. So Victor, he is a molecular microbiologist, but also is very skilled in bioinformatics. Then I want to introduce Guillermo Garrido. Guillermo, yeah. So Guillermo is trained in bioinformatics at the University of Amsterdam. He joined the Leiden University team with Victor, working on bioinformatics projects. So he knows all about, you know, coding, etc. So, and then there is Luisa Arias Giraldo. Luisa is trained in biology, but has adopted bioinformatics as her second, skills. So also she will be helping you a lot. So Guillermo and Luisa with the bioinformatics. So what I did this weekend, first the learning objectives. Yeah, so during these 2 weeks, you will become familiar with the basic concepts and of course also the general trends in microbiome science, because I looked at all your backgrounds and not all of you are in plant microbiome research, also soil science. I saw insects, I saw nematodes. So again, you know, we will talk more on that. So all the things that we will be teaching this week, they are not, you know, special for plant microbiome, they are special for microbiome research. So even if you would work on microbiomes of humans, you could use still the same tools and skills that you will learn in these 2 weeks, okay? Then of course, yeah, the strengths and the weaknesses of high throughput amplicon metagenome data sets. I saw Victor has a beautiful slide. I'm not going to make a spoiler, but you will learn and understand that. Then there's always technical issues that can lead to biases, especially with sequence data analysis. So we will also talk about that. One thing you will not learn is to do coding. Yeah, so we will not learn you to be a bioinformatician. We will learn you how to use it in your work, right? So don't be scared from all the screens that you will see in the coming days. We're not expecting you to do coding, okay? Just how to use it, where to look for, you know, what are the most important points to really look at. So that will be done. So that's the hands-on bioinformatics. And then there is one day where we will discuss literature. The literature discussions has two purposes. First of all, you will look at those papers again with the knowledge that you will gain this week, right? So, okay, what kind of tools did they use to basically address a research question? And the literature also serves to even expand beyond the literature or the more like the content type lectures that we will give on microbiome research, okay? So it will give you a bit of a broader view of what is out there. So that's that. So what I did is the following. So this weekend I made a slide of, I always like, you know, one lecture, one slide, one course, one slide. So the whole course is in this one slide. So the idea here is that, of course, you know, everybody here is working with some sort of a system. I think I should have put soil here as well. There is an insect, there is a plant, there is a nematode here. But again, the study system that you have is equally relevant for this course. Okay, so then today what we will do in the practicals, we will extract environmental DNA, in this case from plant roots, okay? And that's sort of like, that's not what we're gonna do, but I think in your project you will then send that DNA to a company for sequencing, right? That's normally the way to go. And then what is, I understood from Dr. Kumar that many times you get the sequence information back, but usually you ask also the company to do already a data analysis. In my lab and also the lab in Leiden, we don't do that. So we get the raw data from the, and then we start working on those raw data. And it's not that we don't trust the companies, but the companies, they usually use sort of like a super standardized pipeline for many. But your research project usually has very particular, you know, questions or things that you want to look at. And then what we do is we always go back to the raw data. It's not only for sequencing, it's also for metabolomics. So one of Kumar's students, Karishma, she is now in my lab, also working with Guillermo, Luisa, and Victor. She brought genome sequence data and also metabolomics data. And the data that she brought, there was already some processing done. So the first question we had, we need the raw data that come in. Because, and that will also be explained tomorrow, sometimes in these pipelines, information that you get is thrown out. And sometimes there is some valuable information there. So anyway, we will go through that. So having the sequence data that come in, and we have provided a number of files in a shared drive, and we will talk about that later this morning or early afternoon, to set you up where you can find all these things. But the first thing we will do is we will do a taxonomy, phylogeny type analysis. So which microbes are there in your sample, right? And then there is also, if you want to go to functions, you can do a metagenomics type analysis, right? So that's this bar going this direction. But it's from the same sample set, okay? And then there is also, usually you want to pinpoint microbes that are associated with a particular function of your system. And that is called a differential abundance analysis, and you can do that both at the functional level and the taxonomic level, right? So you will learn also how to do that. Then today, because normally the way we work is when you have your sample, you know, part of your sample goes to the sequencing, but part of the sample goes to the more classic microbiology type. So you start isolating microbes from that particular system. So that's culturing. I know some microbes are very difficult to culture, but later on, if you want to validate some of the things that you find in the bioinformatics analysis, at some point in time you will need, to have the microbe in hand, right? So usually you do then phenotyping back. And phenotyping can mean, for example, you can screen for extracellular enzymes or for specific secondary metabolites or volatiles or, anything. And that's also part of this course. Usually with the microbes, you will do the ones that give that particular phenotype, or the consortium that gives that phenotype, you do sequencing of their genomes. And that's nice. What many groups don't do is, when you have that information, actually to connect that work, so the culture-dependent and the culture-independent methods, to connect those. And that's what we call the cross-link. So at the genome level, but also at the functional level, you can connect your microbes with basically your bioinformatic analysis of the entire community. So genome assembly and comparative genomics, we will also do in the course. That's going to be sort of like the menu for the next, I think, 2 weeks. Yeah, exactly, 2 weeks. So what we will do, so again, all these slides, etc., will be made available to you. So no worries there. You don't have to take pictures or whatever. So just, this is all going to be available. With every practical that we do, we will show this slide to you, and then we will tell you where we are going to go in that afternoon practical. Yeah. I think that is, if I remember, yeah. So today we have the introduction, the learning goals, etc. We talked about that now. Victor is going to, at the end of the morning, is going to give 2 lectures. And then early afternoon, it's what we call the check-in, or the baseline. We're going to work with you to see how is the software that you, I think, already installed on your laptop, right? How is it working? And Louisa, she will give sort of like a quick course on R Studio. There will be a small exercise. She will show you a bit on the R graph gallery, how to use that. GitHub, that's a system where you can connect for all these R related program, and then also the link to the Google Drive access. But I think Kumar,
we have already shared it.
We already shared that through the WhatsApp group, right? Okay. And then the rest of the afternoon is the wet lab work, where we do DNA isolation from, I think it's wheat plants, and then culturing from the same sample on plates, right? And then tomorrow we go into the next step of the process. Let's make an agreement. If there are things that are not clear, just ask. Never be afraid to ask. Okay? There's no wrong question. You can always ask. Okay? I'm not sure if we know the answer, but just ask. Okay? So that's how it works. I think with the whole team. We are here to work with you, to learn new things. So just ask. There's really no problem.
That's it. Yeah.
Okay.
Yeah,
Good.
excellent.
Thank you. Wow.
Please come on to the dais.
I request all dignitaries to be on the dais, please. Thank you so much, sir, for that icebreaking. Next, I would like to the students to introduce themselves with their names, their field of study, and their interest in the program, starting from the first row.
Next person.
Greetings of the morning to everyone. My name is Aisha. I am working under Dr. James Singh. My area of research is diversity analysis and characterization of bacterial center of tomato, which is caused by Charlobacter michiganensis, species michiganensis. Actually, the area of interest is like when the training program was started, I was very interested in this. But as I'm not directly working in the plant microbiome, so till last moment I waited and I got this opportunity. I'm hoping for good, I'm hoping for something to learn from this experience. Good morning, everyone. Myself, Juliana. I'm currently working under Dr. Kalyanji Mandal. My topic is on molecular analysis and management of glycolytic unit causing plant-to-glycosides. And I would like to thank Kumar sir for giving this opportunity. Good morning, everyone. Myself, Sunita Patnaik. I'm from PhD third year, and I'm working under Dr. Ravi Gowri. And my work is totally on microbiome, and my topic is determination and exploitation of mesh phylomicrobiome against bipolaris melis causing by southern fern leaf blight. And I give thanks to Dr. A. Kumar sir for giving this opportunity. Thank you all.
Very good morning, everyone. My name is Sudhir Adhikari, and I'm working on plant biome for the identification of new viruses and bubbles. Thank you.
So, Dr. Kumar, all of them are from IIM?
Yeah.
This guys, all of them are from IIM.
Two from IIT Delhi. There's a special request to be selected.
PhD student in plant pathology. I'm working at a doctor's sir. And my work is on characterization of various species and vascular resistant plant.
A very good morning to everyone. I am Vajramma, and I'm going to the second year under the guidance of Dr. Deva Kamil. My work is on characterizing diversity, pathosystem, and genetic exchange studies and detection techniques of alternative leaf and root plots in apple. My work is not directly related to microbiome, but there is a lot of scope from apple leaf plots, which has a lot of from fruit and leaf, which has a lot of biodiversity, microbiome. Thank you for giving this opportunity.
A very good morning, everyone. My name is Natasha Kashyap, and I'm a PhD second year student, currently working under the guidance of Dr. Mirkhan Singh Gujjar sir. My PhD research work is molecular basis of resistance against bipolaris urutiniana causing spot blotch of wheat. So the main focus of my research study is on the transcriptomic analysis in the transcriptomic part. So I'm looking forward for learning new things. Thank you.
A very good morning to all. My name is Poorni Pasha. I am currently working under Dr. Mirkhan Singh Gujjar sir as a MSc second year student. My MSc work is on relevance, identification of resistant sources, and assessment of bioagents against bipolaris solutiniana of small blotch of barley. And I am looking forward for this training program.
Very good morning, everyone. I am Roshni, and I am currently pursuing my master's under the guidance of Dr. T.K. Bhatt. My research topic is biocontrol potential of Weberii bisciana against solvent plant pathogens of chickpea.
A very good morning to all. I am Manoj P.A. and I am an MSc scholar under the guidance of Dr. M.S. Rahan. My work is on Fusarium tetrate of wheat. I am working on the biocontrol of Fusarium tetrate of wheat with the help of the Trichoderma and bacteria biocontrols. So this training will be very helpful to understand more about the microbiome world. So I'm looking forward to it. Thank you. Thank you.
Very good morning, everyone. I'm Gwen Murugan from Kumar Sir's lab. In my MSc, I worked on endophytic baseless mediated suppression of blast disease. Now I'm currently pursuing first year PhD. In my research work, I would like to take on the metabolomics part for my research. So my interest is on metabolomics. Thank you.
Morning, everyone. Myself, Nitish. I'm from microbiology division. I'm doing MSc second year. I'm under the guidance of Dr. Govind sir, and my work is on biocontrol potential of actinobacteria against major rice disease pathogens. Thank you for that.
Good morning, everyone. Myself, Varuna Moitro, MSc second year student from the Division of Microbiology. The title of my thesis work is Exploring Seed Bacterial Microbiome for plant beneficial threats and abiotic stress tolerance. As this is related to this training program, so I am very interested in it. Thank you, ICAR sir, for giving this opportunity. Thank you.
Good morning, everyone. Myself, Sonu, from Division of Molecular Biology and Biotechnology. I am the master student, and my research work is specifically on root modulation, the effect of rhizobium on root modulation in acidic condition. So, this training will be helpful for me to knowing about the more microbial world around the rhizospheric atmosphere. So, thank you, operators, to give me this opportunity. Good morning, everyone. I am Viveka, second year PhD student, working under Dr. Kalyan Kumar Mandal, and my research paper is on function analysis of Zyra gene in bacteria in regulating the reactive genes during biopathogenesis. And as a plant pathologist, I would like to use this opportunity on how this plant microbes will help in controlling the plant diseases. I thank everyone for giving this opportunity. Thank you. Good morning, everyone. Myself, Priya from Division of Microbiology, and I am working under the guidance of Dr. Rajit Kaushik sir. And my title of research work is based on enhancement of phosphorus mobilizing and mobilizing microbial association for enhancing pH efficiency in green bag. And my area of research is like I would like to learn about how to analyze the diversity of culturable and unculturable microorganisms in the particular disease. And thank you everyone for doing this.
Very good morning to all. Myself, Kavya, PhD scholar under Dr. Ramakrishnan, sir. My research work is on functional microbiome of rice under aerobic and flooded conditions. So, most of my work is based on metabolo and metatranscriptomics. So, this will be very useful. Good morning, everyone. Myself, Priya Chaudhary. I am working in the supervision of Professor Shobhit Sharma from IIT Delhi. My work is to understand the general mechanism of disease suppression in organic foods. And thank you, Dr. Kumar, for giving me this opportunity.
Myself, Dr. Govind Sami, CA scientist in Division of Microbiology. I am working on microbial related system. Plus, nowadays we are also started exploring acne bacteria for both microbial and in this training from I like to know more about the biology. Thank you for
Morning everyone, myself Samram Rai. MSc second year from the Division of Microbiology, IIM. As I am working in related to microbiology, so I think this program will help me a lot. Thank you.
Very good morning to all. I am an Osman PhD third year student and working under the guidance of Dr. ICAR sir. And my current my thesis title is Phylogeny bacterial suppression of bacteriocide, and I will be using the metabolome for the suppression of bacteriocide. So this is directly related to my work, and one of the objectives is microbial diversity analysis. So I would like to thank you all, and I am very fortunate that we got the opportunity to learn it from you. Thank you very much.
Very warm good morning to all not present over here. I am Nisha Padar from PhD third year student from the Division of Food Science and Food Service Technology. My work is based on the bacterial volatilome ubiquitous in banana fruits incited by the Coledotrichomycin. Through this training program, I will be learning transcriptomes, general microbiome study that will further assist me in carrying out my PhD research work, and I thank you very much.
Good morning, myself here Amina from Division of Science and Agricultural Chemistry, a second year student, and I am working on the chairmanship of Dr. Kayan Manjaya on the topic of well
survivability of probiotics and to enhance the shelf life, searching ways to enhance the shelf life of symbiotics. So from this workshop, I would like to gain more knowledge on this metabolome study, and thank you for giving me this opportunity.
Very good morning, everyone. Myself, Naishan Prakash, from Division of Microbiomes. I'm currently pursuing my PhD, second year PhD under Dr. K. Soman at Kenya, and I'm working on chickpea meso-rhizobium interaction. So I hope all the techniques and the information which I'm getting from this will be useful for my research progress, and thank everyone for giving such an opportunity.
Good evening, everyone. Myself, Sagar SP. I'm from Dr. Ramakrishna's laboratory, and we are working on microbiome assembly as well as biological chemical biology. And my work is that we will be taking energy, and we are basically working on so looking into the vast data between time metagenomics and the analysis part. It is a little complicated, and I would like to gain information related to analysis part of metagenomics and metabolomics. Thank you.
Good morning, everyone. Myself, Hariappa. I am PhD second year student. I am working on the research of Dr. Meetingabolomics. And my thesis title was Role of Potential Enterprise to Underline Characteristics of Wheat. And all the knowledge coming from here will be helpful for my research work. And I would like to thank Dr. Meetingabolomics sir for giving me this opportunity.
Good morning, everyone. I am Rashi Tyagi, a research scholar under the supervision of Professor Shilpi Sharma at IIT Delhi. And I am working on the bottom-up approach of designing synthetic microbial communities for biotic stress mitigation in agriculture. And I am looking forward to learn the culture-dependent and independent approaches to inculcate them in my own doctoral work. And thank you, Dr. Kumar, for allowing me to attend.
Auspicious morning to everyone. I am Kalaivanan, working under the chairmanship of Dr. Kalyan Kumar Mandal sir on elucidating major gall effects responsible for blight development in rice. So we have been reading so many concepts, tools regarding this microbiome from our PG and PhD. So many dots are there. Hopefully in the coming days, my dots will be knotted. So thanks for the whole organizing team to making me as a part. Thanks so much.
Respected teachers, seniors, my dear friends and juniors. A very good morning. I am Lance Leslie Johnson from Division of Hematology. I am currently pursuing my PhD with Dr. Vishal Somavanshi on understanding the molecular processes modulating the symbiotic relationship of an entomopathogenic Nematotetrarhabditis bacteriopora with its monospecific bacterium Photorhabdus luminescens. So in my research, I will be covering on single-worm transcriptome and functional validation of genes responsible to RNA interference. So here I would like to learn how I can do the bioinformatic analysis from the raw data of a transcriptome data. And finally, I am grateful to be here and thanks for this opportunity.
Good morning, everyone. My name is Wally Bogo. I think I am one of the participants to make this training more international because I'm bringing in a new continent coming from Africa, from Cameroon, with name as Africa in miniature. So I'm here in the frame of a fellowship of the World Academy of Science and the government of India. So I'm working in the research department, research team of Dr. Ekoma. So my research team is metagenomics and culturomic analysis of biocontrol agents for the aflatoxin-free production of maize in Cameroon. So I'm having a very huge project, but I'm from biochemistry division in Cameroon. So I came here to be trained in microbiome and in all the omics knowledge, so that once back in Cameroon, I should be able to train many others also. Omics are very embryonic in our place, so I'm very privileged to be here and to go through this training, and I hope that at the end I will go from the level 0 to level 10, I think. Thank you.
Thank you, everyone.
There's
I
one more
hope
participant in the front. Swarna Lakshmi.
Good morning, I'm Swarna Lakshmi, microbiologist from ICAR. I'm particularly working on regular microbiome, but we are interested on what are the non-rhizobial diversity associated with the nodule tissues and how they help in improving the microbiome function.
Thank you, everyone. I hope this program will benefit you all in your respective research area. With this, I request Dr. Vishwanathan, Joint Director Research IARI, to give some remarks for the audience of the program.
A very good morning to all of you. Professor Jose Raimakers, Dr. Victor Carrion, Dr. Guillermo, Dr. Lisa Maria. I welcome all of you on behalf of the ICAR, and as well as on behalf of the Director IRI to this prestigious Agriculture Research Institute, which is known as the seat of Green Revolution, and today it is the seat of the continued to contribute to the food security of the nation. So we welcome all of you, and from the one slide, I think all the students will agree that Professor Raimaker presented. You will learn a lot, and he summarized the entire course in his excellent way of communication about this training and its importance. So I am really grateful to all of you. I welcome, I am thankful to Dr. Anuradha Agarwal Madam. She is the National Coordinator of the NAHP COST project. She is an eminent scientist who contributed in the area of plant genetic resources, and recently she has taken over as the National Coordinator for the NAHP COST program. And Madam played very key role specifically in organizing this training, especially for getting permission or from the Ministry of External Affairs and various other places. Madam, we are very thankful for that. And besides all your guidance for the entire COST program, we also thank the National Director, Dr. R.C. Agarwal, for his constant support, although he is not available here. The most importantly, the training organizers, Dr. Kumar, Dr. Ramakrishnan, and along with the team from the Leiden University. This program, in fact, Madam, we planned this program in 2019-20, then that time we could not carry out, and finally, with constant effort of both the parties, we could finally organize this training. Actually, we sent about 30 students for foreign training in some or other aspect, and we thought bringing foreign expert here will benefit more students, although all students are from IRI, but they represent mini India. They represent all the almost several states of the country. Fortunately, we have an international participant also. So this is an important aspect. We planned these trainings, and this is the third training in that series. One training we conducted by inviting Professor Ravinder Singh from University of Colorado, and at that time only he came, and three professors from USA, they joined online from their university, but that training was only very short duration, only 3 days he was here and trained. And then we organized a small workshop with the help of Dr. V.R. Gupta and Paul from CSIRO Australia for on the microbiome for the benefit of the student. And this is the third training, and here the entire faculty from Leiden University is here to teach you. So indeed, we could have accommodated more students, but from the other university also, Madam, but for the issues of accommodation, we could not call, and because this training was also postponed few times. But Kumar made an arrangement to live cost this training. More than 100 students registered from different universities in that. It is currently on live, and so they will also get benefit. And I thank all the support provided by Dr. Manjaya, the Nodal Officer Cost Program. I welcome all the students, and these students, our students are we call it a cream of Indian agriculture, because they compete in the national level competition and the top student comes to IRI. So I think really this training is going to be really very useful, and most of them almost this IRI contributes to the agriculture research service, and most of them they will be joining as either the scientist or as assistant professor in different teaching universities. So this training will really very helpful. So about this training, this is we are in the very exciting area of biology today. So when I joined in MSc, if we do a PCR or if we can do a northern to just see the expression of a single gene, it was a great thing. But in 2000, when the Arabidopsis genome sequenced, we were got very much excited, and we thought now we can understand the biology so easily. And if some of you told that you are not directly working on the microbiome, you are working with other aspect, but that there is a paradigm change in the way we think. So most of us, we might have studied in our undergraduation that phenotype is genotype into environment interaction that leads to phenotype. But now the genotype is again divided into two components. One is the genotype of host, and another major component is from the genotype of the microbiome that is present in the host. So in the early 2000, if you see the biologist code would be understand the function of the gene sequence in diverse environment. It was a decadal vision in 2013 when the US academies, they thought this is very important aspect, and they put it in a decadal vision, that is within a decade we will understand function of all the sequence in the diverse environment. But if we see the progress, even we have not understood the function of all the sequence in any sequenced organism. Still we are in that way. Then interactome of those sequences or their sequence product within that organism. Now the more complexity has come in with the introduction of microbiome. So we are in very exciting phase. So now the phenotype is genotype of both microbiome, into environment, into management. So it is a very complex space, but it is a very exciting space for any biologist to work in this area. So you are fortunate to work in this area, and this area provide opportunity to provide more basic work as well as to provide product to the farmers and consumer that are more environmentally safe and production efficient, less cost effective. So I request all the participants to take this opportunity, effectively use this opportunity, and enjoy doing the research. So I think nothing is more exciting than understanding or studying the biology in its large complexity. With this brief remark, I profusely thank Dr. Kumar not only for conducting this training, but he put a lot of effort in coordinating all the training program that we organize for the student, and hope you will enjoy this training. Thank you everyone for sparing your valuable time and coming for this training. Thank you very much.
Thank you, sir. I request the National Coordinator, Dr. Anuradha Agarwal, all dignitaries and our experts to release the training manual.
Thank you.
Thank you, everyone.
The cover design by Victor. So he prepared the cover with a lot of matching images for this book. Excellent.
You also did the
I give the foundation, but you did the final finishing job. Thank you.
Thank you so much. I request Dr. V. Ramakrishnan to propose vote of thanks. Okay, sorry. Now I would like to request.
No, it's a slip, yeah.
Now I would like to request Dr. Anuradha Agarwal, National Coordinator, NAHEP CAS, to give us some remarks on the program.
A very good morning to all. Professor Horst Reimakers, the course director for this course, international course director, and director of Microbial Ecology Institute of Ecology in Netherlands. All his colleagues from Leiden University and NEO, welcome to India and to Delhi. Perfect weather to be in Delhi. And I understand Professor Hose is here for the third time, but I'm sure the others are coming here for the first time. Okay, so make the best of this. Dr. Vishwanathan, the very strong pillar of IRI as Joint Director, and more importantly, the Principal Investigator of the CAST project, a very prestigious project on genomics-mediated crop improvement and management. Dr. A. Kumar, again, the anchor person for this entire training program. He has been interacting me for very long, and there was a point of time when this training almost did not happen, and he was made tremendous efforts to make it go. And now I understand today, Professor Host, from your presentation, why he was very enthusiastic that we make sure that we have this program, in spite of the fact that we are in the middle of the convocation week of this university. It's a week-long affair, very, very interesting program, and the students have taken time out from that and be here. So indeed, I can understand, Dr. Kumar, why you were so enthusiastic.
Yes.
Dr. Manjaya, the very silent nodal officer of the CAST program, but again, a very, very important person for the project as well as for the training program. Dr. Bala Ramakrishnan, microbiologist at IRI and also part of the organizing team of this training program. All the faculty of the department, and of course, most important, trainees. I am so happy to see that this training has become a reality, and we have a cross-sectorial representation of students, very young students to faculty, of course predominantly from IRI and IIT, but I now understand what is Dr. Kumar's strategy is to in one go have this whole team of young enthusiastic researchers getting training in this frontier area of plant microbiome, and we'll have this, like you start from a cell culture and develop a whole colony. So that's the strategy. Congratulations to the entire organizing team. So I would just like to first take a brief moment to introduce you about our NAHEP project, the National Agriculture Higher Education Project, funded jointly by the Government of India and the World Bank on a 50-50 cost ratio basis. So we started this project in 2017-18 financial year, and we are in the last lap of the project. Dr. Vishwanathan represents one of the components of the NAHEP project. So this NAHEP project is about how many? $165 million approximately. which converts to about 1,100 crore rupees. And this basically is aimed to bring transformative reforms to improve the quality and resilience of agriculture higher education by promoting efficiency and competitiveness. And this is being achieved through changes in the working mechanisms of agriculture universities spread all over the country. But we want to raise the teaching and research standards of all agricultural universities. And besides, of course, infrastructure and technical competency. So technical competency training programs like this help a lot. And basically trying to make agricultural education more attractive to students. As Dr. Vishwanathan mentioned, we are already having amongst us the best of the best students in the country coming forward for undertaking agriculture education. How many of you have taken this course, I mean MSc or PhD, as a first choice for your education? Or was it your second or third choice after medicine and whatever, engineering? Anybody would like to raise the hand? First choice to do graduation and post-graduation? So this is the objective of NAHEP. We want to make agriculture education as a sought after career, not as a second or third choice. Because in COVID, we have all seen everything can stop, everything can go under lockdown, but not agriculture and the food chain. So everything in India was under lockdown, strict lockdown, but the farmers were working in their field. That is how important agriculture is. So you people should feel proud you have chosen this field, even if most of you have taken it as a second or third choice. So you have to make agriculture more aspirational by talking to your friends, to your colleagues, and asking and telling them the importance of this subject. And it's cutting edge research, as this training will show you. I mean, there is nothing no new science that is not being applied here. We deal with more than 90 subjects in this field of agriculture education. And in India, we are very, very lucky. We have a network of 74 agriculture universities, state-sponsored, spread all over the country. And the Indian Council of Agricultural Research, which is where our parent organization, is facilitating the education in these universities through core funding as well as additional fundings like NAHEP. So broadly speaking, in NAHEP, we have three components. One is to provide support to these agricultural universities, under which there are subcomponents. One is subcomponent Center for Advanced Agricultural Science and Technology, which is abbreviated as CAST. You will see it on your document. So we have 16 projects under CAST. So these are different centers of excellence in different subjects. Like here it is genomics, somewhere it is secondary agriculture, somewhere it's predicted agriculture, somewhere it's application of drone technology. So all cutting edge research is being funded by the NAHEP, where both students and faculty are benefiting, especially through international exposure like this. And indeed, as Dr. Vishwanathan said, or was it Dr. Kumar, that while many people, both faculty and students, have traveled abroad under this project, it's far more sensible to bring a team like yours and give a hands-on training. So it's in a hybrid mode, right?
Yes, in hybrid mode.
So some physically and some online. In fact, use of ICT technology is one of the major aspects which NAHEP is covering under another component called Component 2, where we are trying to enhance ICAR competency in leadership in agriculture as well as ICT tools. So I'm also looking after that. There we have supported all the 74 agricultural universities to bring to upgrade the infrastructure. We have introduced smart classes. We have introduced many softwares for enhancing teaching experience. E-content has been developed for most of the courses, especially forced during the COVID period. while it was envisioned before the start of the project, the COVID gave it a real push to move towards digital education. In fact, I would like to inform you, we are holding one international conference on blended learning ecosystem on 21st to 23rd March, where we will be discussing how ICT tools and higher education, especially in agriculture, will be the way forward for the next decade or so, and how we need to transition from our current system of education to a more blended system. So I invite you, extend a warm invitation to you if you're planning to come, or if you happen to be in India during that time. I will share the details with you. So this NAHEP project is just to change the entire way we have been teaching and learning in higher education in agriculture. To make it more global, one of the objectives of NAHEP is to convert universities to become more global, get into global rankings like QS ranking. Hopefully IRI will be featuring in the QS ranking very soon, again based on the efforts of this NAHEP project. So broadly, this is what we are doing under the NAHEP project. We are supposed to finish it by March 2023, but since a little budget is left, we might be utilizing it for a little more period of time. So trainings like this is an excellent way of enthusiasing the students and the faculty to do more and more better research, and ultimately with the objective of increasing sustainable agriculture. Because while today we produce enough food, we have produced enough food that for 2 years in a most populous country like India, the government could provide free food to all those below poverty line. That's a huge, huge accomplishment by a country like India, and this large part of it is contribution from the area of agriculture research and development. So feel proud in what we have been doing. You are very lucky to have been invited and selected to attend this training program. And like I said, from the first presentation by Professor Yos, I would like to actually attend the training program. It's going to be an excellent life-turning experience for you people. And not only you are going to make friends amongst each other, you're going to make international friends. And like Professor Kumar, that friendship lasts for a lifetime. So you have been associated for more than 2 decades. That is an enormous thing. And that is something you people should also aspire. I mean, you're now in a WhatsApp group. You will get to know each other. Of course, most of you are IRI students, so probably know each other a little bit. But in these 2 weeks, you will interact more closely and you will get a greater association. Nurture this association for a lifetime. Because again, in research, you would be seeing collaboration is what takes you forward. So keep the collaboration going, have brainstorming between each other to develop new ideas, new concepts, and how you will contribute towards the development of the nation and the globe altogether. Finally, technology, while being important, providing us many tools and pathways to move forward, also sometimes becomes a little, overwhelming. I mean, now ChatGPT answers all our questions. ChatGPT has become co-authors in paper. They have cracked entrance exams. So where are we moving forward? Have a discussion on these aspects also. I mean, technology is exciting and provide us goods and services, but it can also be used negatively. So always keep that in mind. I mean, we are all using mobile phones. More important, it has become more important than any family member, unfortunately. But it also has health hazards. So whatever technology you get into, do a SWOT analysis. So keep that in mind. There was a very interesting article in the Times of India 2 days ago. Please do read that about, it was by Professor V. Ram Gopal Rao, who's the former director of IIT Delhi. Do read that article on ChatGPT. Can't compete with artificial intelligence, but we can stay relevant. So as humans, as soon as machines take over most of the routine jobs that we do, how do humans remain relevant? So while doing cutting edge research in plant microbiome, also have a discussion on those issues. And amongst such a wonderful team of experts, I will not venture to talk about the subject. As Professor Vishwanathan said, I am a person of plant genetic resources, dealing with whole plants and the diversity in them, and how to study them, how to collect them, conserve them, and use them. It's really fascinating that the world of microbiome has such a potential, you know. We normally only like to count the number of plant species, animal species, etc., etc. But I'm sure the microbes put together beat them all in their presence, in their diversity. I mean, they are present even within the cell. I mean, that is quite fascinating of bigger organisms. So, like I said, I would have definitely liked to attend this training program, but Dr. Kumar didn't offer me the option like he did for some of the faculty of IRI. So, all the best to you and to the faculty from Netherlands. Have a good stay in the country. The weather is perfect. Do go around the city. I mean, of course, you'll have many lab hours to spend, but do visit our city. It's a beautiful time. And I'm sure our hosts at IRI will take care of that.
Yes, thank you.
Enjoy the Indian food, which also is very diverse. Thank you. We would like to think so too. And all the best to all of you. Feel free to interact with the faculty. He has already given you an open invitation. Don't be afraid to ask questions. This is a wonderful time. You will remember this training for the rest of your life. And the takeaways from the training are going to help you in your career, whichever pathway you use, whichever field you get into. It will be a lifetime of learning. Make the best of it. And on behalf of the entire NHEB team, including the National Director, Dr. R.C. Agarwal, I extend a very warm welcome to the faculty, international and national, as well as to students. Thank you.
Thank you.
Thank you so much, ma'am. I request Dr. B. Ramakrishnan to propose vote of thanks.
Excellent.
Morning. I am privileged to propose vote of thanks on this special occasion. I'd like to thank all the delegates who blessed us with their presence. I'm more thankful to Dr. Anuradha Agarwal, National Coordinator, Dr. Viswanathan, Joint Director and Project Investigator, Dr. Manjaya, the Nodal Officer, and the team of other members, trainees, and the trainers. Well, for all of you, for your good words and inspirational remarks. Words are not enough to thank the team of Professor Raimakers, Dr. Victor, Dr. Gulimore, and Dr. Lucia, because they accepted our invitation and went through several sessions of online meetings, refined our program, and come out with all the proposed course content and also the lectures. On behalf of the organizing committee, I also thank our regards to Director, Vice Chancellor of our institute, DDG, ADGs, and Ministry of External Affairs and their officials. They helped us in obtaining the administrative approval, but for them, the event may not be possible now. And we know that we have postponed it once and got our approval on time for the actual event today. And my sincere thanks to the NAHP CAST team for supporting us, but for them, I think this event is also, it's very difficult to conduct at the institutional level because it is, I think, after a long years, we have an international team of researchers and professors coming and giving us a training. Otherwise, it used to be one-way flow. We move to their laboratories to learn, rather they come here. And I express my thanks to all the HODs and the faculty members who are online, and also to all my colleagues and who helped us in organizing this event. I thank the faculty members and the committee students and for the staff members who helped us in accommodation, catering, and also on audiovisual programs of today's event. My words are not enough to thank every one of you because you have shown the interest to learn. That is more important to us because unless you show the interest, it won't be possible for us to teach you, and you should be the receptive one to learn from us. I thank all of you for your time. I thank you very much. Thank you very much.
Thank you, sir. I would like to extend a warm welcome to all the participants, keynote speakers, and distinguished guests. I hope that this workshop will be a rewarding and insightful experience for everyone, and that we can work all together to advance our understanding on the topic. Thank you, and I wish you all a productive and enjoyable workshop. Now, I request you all to assemble in front of Discovery Lab for a group photograph, and after that, please have refreshments served near the auditorium. Thank you.
Let's move towards the Discovery Center. The banner is there, so go there and let's take the group photograph. I brought those question paper. 1 page, 25 questions. Take 5 to 10 minutes maximum. No, students. So how are we going to do it? Because it's an examination, right? Sitting closer to each other, your mind might change you. So, so get distributed throughout the hall. Maybe, okay?
You have to.
So remember, you are you are on YouTube. If you are copying, the entire world watches you. All right? So don't do that. Yeah, this is a paper. Who is going to distribute? Elora, please come. Yeah, please.
I will do the second.
Yeah, please. Thank you. Yeah. There will be 25 questions, I think, and the group can distribute. Sri Naina, you can go back. Manoj, you can go back. Whoever is sitting in between, they can go back.
This is for now.
Listen, this pre-training evaluation is not for you. The pre-training evaluation for us. Right? So you will come to know why we are doing this after the training program. Your time starts now. There are 25 very simple, easy to answer questions. Let's see how are we going to do it. Start now. Take 5 to 10 minutes maximum. By 11:50, the paper's back. Everyone got the paper? Yeah, for that. There were 35 copies. Some more people are sitting here. Okay. In the meantime, you can put your presentation.
Yeah.